My name is Roxana Kooijmans, and I will be presenting about my work on quantitative multimodal cell and fiber mapping in full primate brain sections. More than 100 years ago, Brodmann has pioneered work on human brain mapping using Nissl staining in order to compare cytoarchitecture of different cortical areas. More recently, this kind of mapping work has been revolutionized by Axel Schleicher and Karl Zilles, who have created new methods to quantify Nissl staining, such as the gray level index, which has been further developed by Katin Ammons and Timo Dikscheid, who are now generating full coverage cortical maps of the human brain that you can find in the Atlas of the Human Brain Project. My work extends this kind of mapping by making it cell type specific. And this is possible due to the development of a new staining protocol, which has allowed me to map different kinds of cells at the same time in full human brain sections. Here I will illustrate it using calcium binding proteins, albumin, clobindin, and creatinine. Here's a high resolution image of a full human section in the right hemisphere. The section is located about 1 centimeter away from the occipital pole. And as we zoom out, you can see that there are different patterns visible that correspond to different cytoarchitectonic regions. There's this very distinctive blue line that corresponds to a high density albumin positive population in layer 4C of area 17. And as we wrap up, we come back to this very clear border between area 17 and area 18. In the tradition of my collaborators, I am also working on developing a method to quantify these sort of differences. So we are now using machine learning in order to segment cell bodies in these very large images. I'm doing this together with Erik Opschulte in the group of Timo Dikscheid in Jülich. Here is an example of 2 sample regions in area 17 and area 18, and the result of our analysis using a machine learning based algorithm. And I will also show you a detail to see how well the algorithm of Erik is performing. After we have segmented all cell bodies, we are, since we acquire these images using bright field, we have all cell bodies of different colors in the same image. So in order to separate the different populations, we need to perform an analysis which allows us to split this into different channels. And this is actually very easy because the differences in color are very well pronounced, and you can see this in the histogram of counts of different all the cell bodies that we segmented. If you plot the counts of all cell bodies segmented as a function of hue, you can see 3 very distinctive peaks: 1 for blue, which is albumin; 1 for pink, which is clobindin; and 1 for brown, which is creatinine. This staining is also similarly saturated in all 3 channels. So this makes it very easy for us to split the data into 3 different channels. I would like to also show you how stable and reliable this staining is. This is an example of 20 sections from 2 human donors. And these are the distributions of the cell counts in the 20 sections. There are a couple of remarkable things in this slide. The first one is that it contains histograms for about 4 million cells that have been segmented in only 20 images. It is very stable, which you can see in the color distribution. These are segmentations performed on raw images, and yet the location of the 3 peaks is extremely stable across different sections and even across different human donors. It is coherent. You can see smooth anterior to posterior changes in the distributions between the 3 peaks. So the 3 peaks are located at the same position, but they have different heights because that depends on how many cells are represented in each channel, which in turn depends on what sort of distribution they have, what sort of area you are capturing in your section. So we can already see this in these distributions. Going back to my initial example with the sample regions, after we have split our data into 3 channels using this analysis, we can generate maps of distribution in the depth of the cortex, where you can see how numerous the specific populations are along the depth of the cortex and how they compare to each other. And this allows us to map the differences between here area 17 and area 18. A further comparison is possible to also other species when we map them equally systematically. And here is an example comparing to mouse primary visual cortex and macaque primary visual cortex from my earlier work. There is a further extension of this map, which is made possible by my collaboration with Marcus Axer, also in Jülich. Marcus maps the connectome by revealing the orientation of myelinated fibers using polarized light imaging. Here is an example of a full vervet section imaged by Marcus, where you can see the orientation, so you can see where the fibers are going. And I have taken this exact same section, I have unmounted it, and then stained it for calcium binding proteins, which has resulted in this following image. We have then aligned the 2 images to each other, which has allowed us to create this multimodal map. So we are now zooming in to reveal how high resolution these images are, and we have stopped at the lateral geniculate nucleus and the pulvinar, and you can see the very distinctive layering of the LGN. We can zoom in even more. The resolution of both images is about 1 micron, which allows us to see how the different fibers are crossing in the PLI image, and to also clearly distinguish individual cell bodies in the calcium binding protein staining. In the adult LGN of vervet, there's no creatinine expression. It's located in other areas of this brain, but here you can only see calbindin and provalbumin. So if we zoom out again and we follow the optic tract, you can see from Marcus's image how the fibers orient and how they flow towards primary visual cortex. And with the different orientation, we also see changes in the staining, the composition in the calcium binding protein staining, which reveal a subset of provalbumin positive fibers that are included in the optic radiation. So now we have gone back to primary visual cortex and the border between primary and secondary visual cortices as that we have explored earlier. The outlook of this work is generating full cortical coverage maps of the human brain, which is possible due to the fact that we can work with full sections and we have a very scalable method of analyzing our sections due to machine learning and high performance computing. We can perform cross-model analysis of connectivity and cell type distributions due to the fact that we can image both fibers and specific cell types in the same tissue. And of course, we cover the entire brain, so we have a lot of data to compare. And another very exciting possibility is to map individual fibers in the polarized light image and in the immunohistochemistry scan, which is possible due to the fact that we have extremely high resolution images of both stained and unstained fibers in exactly the same tissue. And we can use machine learning in order to do very high level alignments. And I would like to thank all my collaborators in Jülich and in Amsterdam and also in Maastricht.
My project is about dynamically accurate spatial and megapulse MRs for detection of C13 labeled cerebral glucose metabolism. And this project has been done in your lab under supervision of Dr. Jenni. Glutamate and GABA are the main excitatory and inhibitory neurotransmitter in the brain, and glucose, which is the main substrate for cerebral energy metabolism, is served as a metabolic precursor for both glutamate and GABA synthesis. NMR combined with infusion of C13 labeled substrates such as glucose can be used for detecting the metabolism of these two metabolites. For example, when glucose is labeled at position 1, the label enters to the TCA cycle, and glutamate will be labeled at position 4, and GABA will be labeled at position 2. And when glucose label enter to the second and third turn of TCA cycle, glutamate will be labeled at position 3 and 2, and GABA will be labeled at position 3 and 4. Detecting the label and turnover curve can be done either through direct C13 MRs or indirect C13 MRs through detecting the signal from proton who's attached to the C13 carbon. However, these two techniques are hampered by technical challenges because they both need complicated RF coil and also filter for proton and carbon, and a console can handle both proton and carbon frequency. In 2004, Bumesber and his colleague, they introduced a simple approach called dynamic proton MRs for detecting the C13 labeling of metabolite. This technique is based on the pronounced effect of carbon-proton coupling on the proton spectra, and it bypasses the need for carbon-13 hardware and complicated acquisition sequence. And also its application has been well demonstrated in monkey brain. Here I will briefly explain this method. So in each metabolite in the brain, we have 99% C12 and there is only 1% C13. So here you can see the simulated spectra from proton on coupe to C12. And as we expected, there is no visible signal from proton coupe to C13 nuclei. And the MR signal we detect is the sum of these two signals from proton to attach to carbon-12 and carbon-13. However, you can see this MR signal is pretty similar to the signal from proton who's attached to carbon-12. So just if in the beginning of infusion we don't have C13 in for metabolite. So if we subtract the post-infusion spectra from pre-infusion spectra, the labeling spectra will be flat line. But when we started infusion, C12 nuclei will be replaced by C13, and it results in reduction in the signal from proton coupe to carbon-12 and increase in the signal from proton coupe to carbon-13. So now if we subtract the post-infusion spectrum from pre-infusion spectrum, we have this pattern for labeling. So with time, the amount of signal in this small satellite peak increase, and also this negative, the amplitude of this negative peak will increase with infusion time. So the aim of our study was to implement this simple technique in a clinical scanner and investigate the feasibility of this technique and perform the dynamic proton MRs using a special sequence to detect the labeling of glutamate from labeled glucose and perform the dynamic proton MRs using megapares J difference editing sequence to detect the labeling of glutamate and GABA from labeled glucose. So for a special sequence, we use a special sequence that has been implemented in our 3 Tesla scanner. And we use a very short echo time of 8.5 millisecond, and OVS and vapor model was used for out volume suppression and water suppression. And 2 subjects has been studied, 1 male and 1 female. And the voxel of acquisition was positioned over anterior cingulate cortex, and the next voxel position over preconus posterior cingulate cortex. So for MRS acquisition protocol, we acquire 1 MRS spectra just before infusion from both voxel, and then we started our infusion. And MRS acquisition was interleaved between 2 voxel. For megapares J difference editing sequence, the reason that we use this sequence because GABA peak at 3 ppm has overlap with dominant signal such as creatine, and megapares J difference editing sequence is one of the widely used method for separating GABA peak from creatine. So echo time that we use in this sequence was 68 millisecond, and we also done this study at 3 Tesla scanner, and 1 subject has been studied. The voxel position over the preconus posterior cingulate cortex, and we use the same infusion protocol and MRS acquisition protocol for this sequence as well, like what we done for a special sequence. So we, for the pre-processing MRS spectra, we use FIDE toolkit that has been developed in our lab. And S model was used for quantification of the fully processed difference spectra using in-house simulated basis set. We generated 2 LC model basis set, one the pre-infusion basis set that include only the spectral shape of carbon-12 bonded proton signal present in the baseline spectra, and the other one was a labeling specific basis set that took into account the pronounced effect of carbon-proton scalar coupling on the observed proton spectra in a special and GABA-edited megapares sequence, and used for quantifying the labeling difference spectra. So here is the result for the special sequence. You can see in the left side the overlay of the proton spectra acquired from both voxel at each time point before, during, and after infusion. And when we subtract this spectra from baseline spectra, we have this labeling in difference spectra. So we can see that there is an elevation in glucose peak at 3.76 ppm and 3.41 ppm, and the labeling of glutamate at position 4 can shown here by red column. So there is a reduction in the signal from C12 bonded proton at 2.35 ppm, an increase in satellite peak from C13 Coop proton at 1.8 and 2.9 ppm. And also here you can see the labeling of glutamate at position C3 that has been shown by blue column. So here you see one of the example of LC model fit for different labeling spectra. So we can see the individual fit for glutamate labeled at position 4 and also glutamate labeled at position 3. So this figure shows the estimated time course of C13 labeling for glutamate labeled at both position in both voxel. So 90 minutes following infusion of labeled glucose, the fractional enrichment of glutamate at position 4 was 30%, and fractional enrichment of glutamate at position 3 was 4 to 5%. And here is the result for the megapares sequence. So when we subtract the post-infusion spectra from baseline spectra, we have this labeling difference spectra, and we can see obviously the labeling of glutamate by reduction in the signal from C12 banded proton and increase in satellite peak from C13 banded proton. So again, there is here is one of the example of LC model fit of the label difference spectra, and we show the individual fit for glutamate labeled at position 4, 3, and 2, and also GABA labeled at position 2 and 3. So here is the fractional enrichment time course for total glutamate, total label glutamate, and also GABA. So 90 minutes after infusion, we have 27% enrichment for glutamate and only 8% enrichment for GABA. So as a conclusion, we in this study, we demonstrate the use of both two sequence, special and megapress editing sequence to follow the fate of C13 label from infused label glucose in the human brain in the absence of complicated hardware and heteronuclear decoupling arc pulses. And the presence of the C13 label uptake was clearly detectable in metabolite, especially glutamate, and this owes to the pronounced effect of carbon-proton coupling on the observed proton spectra. And fractional enrichment of GABA was 30% of fractional enrichment of glutamate, and this is in contradiction with reported results from previous animal study that reported the fractional enrichment of GABA is similar to glutamate. And one of the explanation for this can be there is a possibility of macromolecular contamination in GABA baseline spectra that has been used for estimating the metabolite pool size, and also this has been reported in a previous study. And then I would like thanks for your attention, and also I thanks to all people in my lab who helped me for this, especially my supervisor, Dr. Jenni.
Hello, everybody. Today I'm going to talk about our recent findings regarding simultaneous mapping of T2 star and major neurotransmitter using spectroscopic imaging at 3 Tesla. GABA and glutamate are the major neurotransmitters, and we all know that the GABA controls the inhibitory activities, whereas glutamate controls the excitatory activities, and they are both strongly implicated in functional changes in neural circuitry. At the same time, GABA and glutamate plays an important role in controlling of the constriction and dilation of the blood vessels, which results in blood oxygenation level, dependency response, or fMRI signals, or T2 star relaxation changes. Now, the bold response and T2 star change can be observed during the activation, functional activation. And compared to the baseline condition, at 3 Tesla, T2 star might increase up to 6%. And this signal not only seen on the water signal, but also at the metabolite levels. In early studies in 2001 from the Minnesota group, Professor Wei Chen has observed a significant signal increase in metabolite levels compared to the control condition. So when they subtract the task condition from the control, and they observed signal increase in creatine and signal increase in NAA. However, this signal increase is not originating from the concentration changes. It is originating from the bold response change or T2 relaxation increase due to the bold response. So this problem becomes more significant at longer echo times. The change in T2 stars exacerbates and results in signal alterations of neurotransmitters due to the bold effect. So it is really essential at this level to monitor the T2 star while measuring the neurochemicals or neurotransmitters. So in this study, we aim to map T2 star with neurotransmitters at the same time. This will provide useful information on functional hemodynamic changes due to the physiological interventions. To do that, we propose to use megasemi laser localization. However, in this case, instead of suppressing the water, we use non-water suppressed metabolite cycling. This allows us to measure the T2 star together with GABA and glutamate, and using the spectroscopic imaging, two-dimensional, we can map these two metabolites and T2 star throughout a whole axial slice. We recruited 4 subjects, and MRI size measurements were conducted at 3 Tesla Siemens Prisma scanner. And megasemi laser with an echo time 6 to 8 milliseconds were used for GABA and glutamate detection. And after the post-processing of the reconstruction data, case-based data, we ended up getting 7.5, 7.5, 20 millimeter cube nominal voxel dimension. All data, including the spectroscopic imaging spectra and metabolite maps, were stored in NIFTY format that allows us to use FSL, FSLIs, SPM, and AFNI for visualization and also pre-processing steps to quantify the metabolites and T2 stars. LC model with internal water reference were used to quantify the metabolite levels. Unsuppressed water signals were also used to generate T2 star maps using the Dehghani software, which is an echo-dependent analysis of multi-echo functional magnetic spectroscopic imaging data. Whereas in this case, we are using the spectroscopy data with the same purpose. This is an illustration of the use of FSL and NIFTY for spectroscopic imaging data and also metabolite maps. Before going through the in vivo experiment, we conducted a phantom experiments with a nominal voxel 7.5, 7.5, 15 millimeter cube. In this case, slice thickness were reduced to show that our sensitivity is better. And we use two spheres with GABA concentration, which are 1 micromole per gram and 2 micromole per gram. The rest of the acquisition parameters were identical to the in vivo acquisition. So we used two averages and total acquisition time 512 seconds. As illustrated on the figure here, you can see we can detect the GABA signal from both spheres, which are 1 micromole per gram and 2 micromole per gram. They are highlighted as blue and green, and blue and red. And expected chemical resonances of GABA resulted from our acquisition and post-processing steps. Here, we used metabolite cycling as compared to the water suppression, which allows us, this scheme allows us to do the voxel-wise frequency, phase, and eddy current correction. Compared to the water suppression methods, which are lack of signal, this improves the signal-to-noise ratio drastically. Also, metabolite cycling significantly reduces the sideband artifacts. Since we are not suppressing the water and we are using the NIFTY format, we were able to facilitate the FSL tool for the rigid body motion correction. And multi-echo information, since we are not suppressing the water, can be used for T2 star mapping. This is an example from 4 subjects, echo time dependent analysis of multi-echo magnetic resonance spectroscopic imaging data from 4 subjects. We also conducted series of anatomical investigation. According to this, gray matter fraction is significantly correlated with the T2 star estimation of the Dehghani tool, and this is consistent across 4 subjects. Further on, we figure out that the gray matter concentration, gray matter T2 star is significantly higher compared to the white matter T2 stars, and which is in line with the literature as well. Since we are not suppressing the water and we acquire 8 unsuppressed water signals throughout the mega semi-laser localization, this allows us to make a time series of the T2 star change during the mega editing acquisition. And this is a quite useful information to monitor how T2 star change while we are measuring the neurotransmitters. Here on the right side of the data, you can see the LC model results overlaid on the MPRH data for glutamate and GABA concentrations. On the left, you can see which spectra were used to quantify these metabolites. As you can see here, we applied an altitude regularization to suppress the residual lipid. However, you can obviously see that this is lipid suppression method is acquisition trajectory is successfully suppressed the residual lipid. However, using altitude regularization minimizes this leakage of the lipid from the lipid trail as much as possible. The final figure that we are going to present in this presentation is GABA, glutamate, and T2 star maps together with the gray matter fraction as well. On the left, you can see the glutamate, GABA, T2 star, and gray matter fraction figures. When we correlate the T2 star maps with glutamate and also GABA, we ended up getting significant correlation for all subjects. However, glutamate signals is correlation with T2 star is stronger than the GABA versus T2 star maps. In the end, as a conclusion, we developed a sequence that generates simultaneous GABA, glutamate and glutamine maps, and T2 star within a clinically feasible acquisition time of 8.5 minutes with a nominal voxel dimension of 7.5 millimeter cube at 3 Tesla. Simultaneous measurements of T2 maps with neurochemicals is a powerful tool to provide useful information on physiological interventions which aim to manipulate excitatory and inhibitor balance. Thank you so much for your attention. My email address is outlined below. Feel free to contact me for further questions. Thank you so much.
Good day, everyone. I'm Kim Desmond from CAMH, the Center for Addiction and Mental Health at the University of Toronto. It's my pleasure to present the work of Sofia Chavez and myself on mapping quantitative magnetic resonance imaging parameters in the brain. The macromolecular pool fraction, or MPF, is a quantitative measure of the fraction of MR invisible protons associated with large immobile molecules relative to the protons in free water that contribute to the MRI image. It is between 5 and 20% in human brain tissue. When one is interested in 3D mapping of MPF, traditional methods are too slow for practical clinical use. Therefore, faster methods have been developed, which greatly reduce the number of required images by fixing invariant parameters during fitting and synthesizing the reference image instead of taking the time to acquire it. We have further improved the synthetic reference MPF protocol by removing a limitation on the MRI sequence parameters to make the protocol even faster. Magnetization transfer contrast and MPF, the quantitative parameter derived from it, have found utility in many neurological applications. MPF is significantly reduced in the shaking pup model of dysmyelination. Histology from a cuprizone demyelination study showed strong correlation between MPF and myelin staining. Variations in human MT contrast over the lifespan in both white and gray matter are consistent with changes in myelin concentration with age. Reduced magnetization transfer measures are observed in a host of neurocognitive and psychiatric disorders, thought to be indicative of reduced synaptic density, neuromelanin, and of course myelin. In the clinical setting, there is insufficient time to acquire the full dataset required for MPF fitting. Therefore, the measurement is reduced to the magnetization transfer ratio, one image produced with MT contrast compared to a reference image without MT. But measurements made solely on the size of the MT effect, such as magnetization transfer ratio, or MTR, are not quantitative, because the MT effect is not just related to MPF. It's also dependent on the sequence parameters, other tissue properties, and inhomogeneity in the transmit field B1. Yarnik studied the dependence of the MT spectrum on these physical parameters and concluded that the offset frequency could be chosen so that the MT contrast is sensitive to MPF and the sensitivity to other parameters is minimized. Therefore, these parameters could be fixed with minimal effect on the MPF estimate. Using these methods, if maps of B1 and T1 are available, only 2 additional images are required for MPF mapping, an MT-weighted image and a reference produced with no saturation pulse. These last 2 are the same required for the MTR, but for this choice of sequence parameters and accompanying fixed parameters and parameter maps is able to uniquely determine MPF. This is much faster than having to acquire images covering the full MT spectrum. Yarnik went on to propose that further accelerations are possible, considering that the reference image has no saturation pulse and hence no MT effects. It can be constructed from prior knowledge of B1 maps, T1 maps, and the map of the equilibrium signal intensity S0, which can be obtained from the T1 weighted images used to make the T1 map. This approach is called the synthetic reference single point MPF. Now, the only additional image required to obtain MPF is the MT-weighted image, cutting the MT part of the protocol by half. This can be appreciated by looking at the block equation model for the spoiled gradient echo signal S. The synthetic reference is fully determined by sequence parameters and variables determined from the three maps. In the previous work, there was a restriction on the scans used to create the synthetic image. The images needed to be acquired with the parameters matched as closely as possible to the MT-weighted image. This is because the S0 image is composed of several factors. The proton density, coil sensitivity, and T2 star are constant, but there is an additional factor, F, which changes depending on the sequence parameters. This factor F includes zeta, a factor related to scanner adjustments, and sigma SP, which is related to the spoiling time. Both change with TR. Previously, the workaround was to acquire the T1 mapping images with the same long TR as the MT-weighted scans, causing an extension to the total scan time. In this work, we wanted to investigate whether fast T1 and B1 methods without matched TR could be used for the synthetic MPF method. We hypothesized that although factors in S0 were changing between sequences, it would change by a predictable constant amount. Once this constant was empirically determined, its inverse could be used to scale the synthetic reference. We call this the calibration factor, F. To determine the value of this calibration factor, we investigated the scale difference between the actual MT reference image and the synthetically generated reference made using our fast T1 and B1 maps. 5 healthy people were scanned with this protocol on our GE Discovery 750 at 3 Tesla. I would like to call attention to the differences between the MT scans and the T1 scans. T1 and MT both use fast gradient echo, but the TR for the MT scans is much longer to accommodate the saturation pulse. Acquiring our T1 maps with a short TR shortens the protocol by several valuable minutes to achieve MPF mapping in 10 minutes and 38 seconds. We created an image which was the ratio of the synthetic and acquired reference, and made a histogram of the value of this ratio at each voxel. The peak of this histogram, the mode value, was recorded for each subject. The mean ratio value across all subjects was 12.64. Importantly, this value did not change substantially between subjects, with a coefficient of variation under 0.5%. We scaled the synthetic reference by this value for each subject, and then compared the MPF derived from the acquired reference to that using the scaled synthetic reference. The MPF was computed from the T1 map, B1 map, MT-weighted, and MT reference images using a fast analytic algorithm, which will be presented later this year at the ISMRM conference. The first column is 2 slices from a 3D MPF map made using the acquired reference, and the second column is the MPF calculated from the synthetic reference. The 2 images are very similar, as expected, with a slight loss of contrast in the MPF map from the synthetic reference. Gray matter and white matter masks were computed using FSL tools, and the relative difference between the 2 cases was calculated. We found that MPF is within about 2% in the white matter regions, but is overestimated in regions where MPF is low, i.e. in the cortical and subcortical regions. In order to examine the origin of the differences between the acquired and synthetic MPF measurements, we looked at error propagation to determine the consequences of error in the B1 and T1 measurements on the calculated value of the MPF. These plots simulate the relative error in the MPF calculation for errors in the B1 measurement. We were interested in the difference between the error propagation using the acquired reference in solid lines and the synthetic reference in the dotted lines. We examined a true MPF of 0.12, close to the value in white matter, and an MPF of 0.07, which is close to the MPF value in gray matter. We plotted the error in MPF for a range of B1 values expected in brain between 0.8 and 1.2, and for the range of T1 values expected in white matter between 800 and 1,000 milliseconds, and in gray matter between 1,000 and 1,300 milliseconds. For the acquired reference, the results are similar for both true MPF values. And for the synthetic reference, the magnitude of the error is greater for gray matter than in white matter. The MPF is most sensitive to B1 errors when the true B1 is low. For the synthetic reference, if B1 is underestimated, then MPF will also be underestimated. Consider that B1 is lower in cortex than in the center of the brain because of wave propagation effects. So there is a maximum difference between the acquired and synthetic MPF when B1 is underestimated in regions which already have low B1. We also looked at how errors in the T1 map would affect the MPF result for different values of the true T1 and B1. As can be appreciated by the overlapping lines, within each case, the error in MPF as a result of T1 errors is similar regardless of the true T1 and B1. But as on the previous slide, the difference between the MPF from the acquired and synthetic reference is greatest in the gray matter. Notably, the error in MPF is less in the case of the synthetic reference. This is due to a compensation effect. T1 is used directly in the calculation of MPF and also in the calculation of the synthetic reference image. These factors have opposite effects on the MPF estimate, so the T1 errors balance themselves out. In conclusion, we have accelerated the protocol for single-point MPF with a synthetic reference by using fast methods for T1 and B1 mapping, making it possible to obtain quantitative MPF in approximately 10 minutes. This required correction by a calibration factor, which only needs to be determined once per scanner and then can be used for all future scans. We compared the MPF calculated in two cases. One, when the reference image was acquired, and two, when the reference image was generated synthetically. Within the white matter, the MPF in the two cases is very closely matched. However, in the gray matter, MPF is overestimated. Error analysis indicates that synthetic MPF is more sensitive to errors in B1 than T1. Therefore, the immediate future work will focus on resolving B1 errors. I would like to acknowledge the support of the MRI group at CAMH, especially my co-author, Sofia Chavez, who developed the FAST T1 and B1 protocol. I would also like to extend thanks to Tobias Wood from King's College London for sharing a copy of his MR Pulse sequence through the GE collaboration portal. Thank you.
Okay, today I'm going to present our abstract on short echo time fMRI using magnetization transfer contrast. The presentation is somewhat different from the submitted abstract, which we used as a pilot study for the full study that I'm going to present here, along with a new title, Arterial Blood Contrast Enabled Magnetization Transfer, a novel MRI technique for enhancing the measurement of brain activation changes beyond T2 star. With this technique, there is no need for the bold contrast to develop to obtain brain activation. Brain activation studies have been the working horse for our understanding of the brain and are used in preoperative planning and diagnosis of, for instance, mental illnesses and neurological disorders. They are usually performed using the bold contrast, which depends on local changes in blood flow, blood volume, and metabolic rate. Disadvantages of this technique are that it uses long TE, which limits efficiency, reduces signal, and is prone to artifacts and dropouts. Furthermore, it relies on changes in deoxyhemoglobin in capillaries and post-capillary vessels, which means that we always measure activation downstream of the neuronal activity. Also, there is sensitivity variation throughout the brain depending on local value of transverse relaxation time. Therefore, people have been looking into alternative techniques to sensitizing the MR signal, which is also the goal of our work. Increasing interest has developed in techniques utilizing CBV changes, as they promise to be better localized to the source of neuronal activity. One approach that is sensitive to changes in blood volume is the use of magnetization transfer. Earlier work that used magnetization transfer to measure brain activation looked into the effect that MT has on bold and showed that MT could be exploited to measure blood volume. However, lengthy off-resonance pulses were used and sufficiently long TE to generate either a bold contrast or attenuate venous signal. Here we propose a different approach to obtain the contrast, which is additionally additive to bold. In our work, we concentrated on minimizing TE and the use of on-resonance pulsed MT. To increase efficiency by eliminating off-resonance pulses and increase sensitivity to changes in blood volume. As magnetization transfer suppresses tissue signal while the blood signal stays largely unaffected, we expect an increase in total signal due to an increase in blood volume, an increase in blood oxygenation level within capillaries, and an increase of water exchange, leading to an increased attenuation of the venous signal. We cannot claim that what we measure at short TE is only coming from changes in CVV, but from the literature we know that more than 60% of MT-weighted signal variation comes from the arterioles, while less than 10% comes from the venous department. Therefore, we decided to call this contrast arterial blood contrast. ABC has the highest signal at TE equals 0. Therefore, an estimation of the pure ABC signal is technically very challenging. We, however, expect that with the introduction of ABC, the activation at short TE will be modified, and decided to investigate ABC in addition to BOLD with a short TE multi-echo grain echo EPI sequence. We designed the sequence in the following way. We start off with a net 0 degree on-resonance MT pulse, consisting of two non-selective binomial RF pulses back-to-back, reducing on-resonance signal attenuation, followed by this pseudo-random gradient spoil scheme to avoid coherence pathways associated with a signal excited with the MT pulse, followed by the actual main sequence. To achieve constant steady state of continuous signal suppression, the MT pulse is played out before every excitation. Therefore, it is not straightforward to interleave the MT experiment with a standard multi-echo grain echo EPI for comparison in one go. We decided to compare what we call our MT on experiment to the MT off situation, where the MT pulse is turned off while all other parameters are kept constant. As scanning protocol, we chose standard fMRI parameters, with the first echo as short as possible and the last to ensure that we get a strong standard BOLD signal in the MT off case, while the TR is below 2 seconds. We optimized the MT flip angle in a preliminary experiment to maximize the signal difference between CSF and tissue with respect to the SAW limitations. The image quality at different TEs shows a uniform suppression of gray matter signal with a suppression factor of approximately 60%. In the study, we acquired data from 16 subjects on a 3 Tesla Prisma system. Using a simple visual stimulus paradigm with a hemifield right-left checkerboard flickering at 4 Hz and a block design, where participants were asked to fixate a gray cross in the middle of the screen during a task duration of 10 minutes. The experiment consisted of 2 runs, one for MT on and one for MT off, which were counterbalanced across subjects. To verify the hemodynamic convolution, we calculated HRS using first-level analysis parameter estimates and found similar temporal evolution for MT on and MT off. We therefore concluded that we could use standard analysis methods for the evaluation of this experiment. In the mixed effect group level analysis, the signal activation for all 3 echoes, we saw significant activation for all 3 echoes in the MT on case, where the first echo shows the highest ABC contribution, reducing with TE while the BOLD signal builds up. In the MT off case, as expected, we could only find activation at TE equals 28 milliseconds, which is a strong standard BOLD signal. We used this as a gold standard and used it to define our RIs to measure our mean Z-scores as a measure of functional sensitivity and the total number of significant activated voxels. For MT on, the highest Z-score is at shortest TE, and we see approximately the same number of activated voxels and similar Z-scores for all TEs, whereas for the MT off case, we see a classical bold behavior with a high number of activated voxels. We also compared relative sensitivity of empty on and empty off. Two interesting results are shown on this slide. At shortest TE, MT shows an overwhelmingly high activation despite the elimination of more than half of the native signal through the application of MT. But also in some regions, bold shows a higher activation than the first echo of the MT on acquisition. Having this in mind, we looked at contour maps superimposed on the early visual cortex. We could find that for MT on, the activation, the Z-scores are tightly localized, whereas for MT off, they expand. Also giving rise to the pattern of the activation as a function of TE, which we've seen on an earlier slide. When we then examined individual beta values as a metric for the activation strength, we could find a narrower distribution for MT on, so a lower intersubject variability, which is responsible for the higher Z-scores that we obtained for the first echo of MT on. Finally, we can say that we found convincing evidence for an additional underlying contrast mechanism, which is tightly localized and enables acquisition at short TE, increasing sensitivity and efficiency, eliminating signal dropouts. The ability to acquire a short TE will give more freedom in the choice of the actual acquisition sequence. So new possibilities for instance, spirals or 3D PI will arise. The final point I would like to make is that ABC is complementary to bold. It is straightforward to implement and to use. So we do not see any reason why this should not be used at 3 Tesla as a standard method and expect it to achieve widespread use in the near future. Thank you.
Hi, everyone. Thank you for taking your time for this presentation and for this opportunity. So I will present my findings on time-of-flight MRI-derived probabilistic map of each major cerebral artery. Bold fMRI is a commonly used hemodynamic sequence to study the brain. It is known to be sensitive to changes in cerebral blood flow and increasingly known to be influenced by vascular morphology. If we take the example of Vigneault and Guat in 2014, we note that regional differences in underlying venous density can have significant impacts on the bold signal. So if we look at the figure here on the left, we see that before controlling for venous density, and in a figure on the right, we see after controlling for regional variations in venous density, we see that there's a significant attenuation of the bold signal, particularly in regions such as the insula and the posterior cingulate cortex. This suggests that it is particularly important to take in consideration underlying vascular morphology. In terms of cerebral arteries, they've received little attention in terms of their effect on the bold signals as of yet. However, they may have an important impact. The brain is supplied by 3 major cerebral arteries: the anterocerebral artery, which territory is outlined here in blue, the middle cerebral artery, whose territory is outlined in yellow, and the posterior cerebral artery, whose territory is outlined in red. Being able to subdivide the brain into each of these regions and take in consideration the underlying characteristics of each artery may be able to give us additional information when we're trying to interpret our fMRI data. So this leads us to the goals of this presentation. First, we wanted to generate a probabilistic map of each major cerebral artery. We then wanted to characterize the vascular density, diameter, and cerebral blood flow within each of the territories, and then show applications of our probabilistic maps in ROIs within the same arterial territory, as well in various regions throughout the brain. So in order to do this, we took a multi-modal approach. First, we obtained a whole brain multi-chunk high-resolution time-of-flight angiography, which you see in the figure here on the left in red. We then obtained a pseudo-continuous arteriospin labeling, again, whole brain, which was quantified using Oxford ASL and shown on the figure on the left and right. We then obtained an anatomical T1 weighted image in order to register our ASL and TOF data into MNI space, as well as control for variations in tissue density within each region. All of this was collected in 9 healthy participants. In order to create our probabilistic maps, we first segmented our time-of-flight images in native space, and then transformed these images into MNI space. So what you see here in the figure is a projection of one participant's segmented time-of-flight image in MNI space. We then manually annotated each cerebral artery separately for each participant. So what you see in the figure here at the bottom is one participant's annotated middle cerebral artery. We performed this for each artery and then averaged across across our 9 participants for each artery. What we obtained was probabilistic maps, which you see here on the left. Above, you see the anterior cerebral artery. Here you see the middle cerebral artery, and then at the bottom you see the posterior cerebral artery. Regions in red are regions where we had all participants' annotated boxes overlapped, whereas in regions in purple is where we had lower overlap across our participants. So what we can note is that within each territory we have variations in the vascular profiles. If we look at the size of each territory, we see that the middle cerebral artery had the largest territory, which is almost double that of the posterior and anterior cerebral arteries. And the posterior cerebral artery had the smallest territory. When we look at vascular density, which is the percentage of voxels within our entire territory that are considered to be belonging to an artery, we see that the posterior cerebral artery had the highest vascular density across each compared to the other 2 arteries across each tissue type. When we look at arterial diameters, we see that, which were extracted in native space. Here on the right, we see a projection of one participant's arteries. In white, we see regions of higher arterial diameters, and in red, we see smaller arterial diameters. We see that while the PCA had much higher vascular density than the other arteries, it does not stand out in terms of arterial diameter, suggesting that this higher vascular density is likely due to increased branching compared to the other arteries. Now, when we look at cerebral blood flow, we also see that compared to the other arteries, the posterior cerebral arteries region had a much higher cerebral blood flow. We believe this is likely related to the increased vascular density observed in this region as well. So if we compare what we've noted, we see that each of the arteries' territories are unique with varying distributions of vascular profiles within them. We noted that while the MCA had the largest territory, it had the smallest diameters, and while the posterior cerebral artery had the smallest territory, it had the highest vascular density and cerebral blood flow. So one thing that's important to note, and that I previously mentioned, is that our arteries, there's a variation in the probability of the presence of an artery throughout the territory. Some regions have a 100% probability in which all of our participants had an artery or a branch going through this region, while other regions in the territory had a much lower probability, showing that some people had different branching patterns. When we compare this to the traditional territory maps of the arteries, we see that they're more distinct regions for each artery, which may not necessarily be accurate, as we may see differences throughout that may not take into consideration if not. So in order to further explore this, we wanted to examine 2 ROIs located within the same region. For this, we used language ROIs, as these are very well studied, and we know that they lie both within the middle cerebral arteries territory. And then in order to explore throughout the brain, we use the default mode network, which is a very popular resting state network, and its nodes are also located throughout the brain, allowing us to explore more thoroughly the effects of vascular density throughout the brain. So in order to obtain our language regions, we first went into Neuroscience meta-analysis keyword search and searched for language. We downloaded the following map, applied thresholding and clustering, and backchecked our ROIs, and obtained approximately Broca's ROI shown here in white and approximately Wernicke's area shown here in black. We then intersected these ROIs with our vascular probability map of the middle cerebral artery. So in this image here, showed by the pink arrow, we see our approximately Broca's area, and shown by the green arrow, approximately Wernicke's area. What we know is that Broca's area resided in a region where many participants had a branch of the MCA passing through, thus resulting in a quite high vascular probability of around 60%. Whereas Wernicke's area lied more on the edge of the middle cerebral arteries territory with a much lower vascular probability, somewhere around 30%. This is particularly interesting as we know it is very well established that both Broca and Wernicke's area are supplied by the middle cerebral artery, and yet when we look at these ROIs, we see that there's a different vascular profile. Now, when we look at the default mode network, we did like with the language ROIs, we went into Neurosynth and searched for the default mode and downloaded the following map. We applied clustering and then selected our nodes of the default mode network, which you see here in the figure pointed out with the white arrows. We then intersected these ROI, each of these nodes, with our vascular probability maps of each arteries, and these are the following results. So what we see is that the nodes of the default mode network fall primarily within the MCA and the ACA's territory, and in regions of very high vascular probability. But we know that at least 3 of our nodes fall within a region of 80% probability. This is particularly interesting when we consider the structure of the MCA and the ACA. Both of these arteries bifurcate from the internal carotid artery. So if we look at the figure here, the internal carotid artery comes up, branches up into the anterior cerebral artery, which goes forward, and then branches left and right into the left and right middle cerebral artery. This is particularly interesting as the blood, since they both bifurcate from the same source artery, the blood flow through these regions is all connected. This may suggest that in addition to well-studied functional connectivity, there may also be a certain amount of vascular connectivity within these regions, which may need to be further studied in future experiments. So in conclusion, what we've shown is that our major cerebral arteries have unique vascular territories with varying probability of arteries. So we have regions of high and low probability within the same territory. We see that ROIs within the same vascular territory can have different vascular profiles, showing this variability in the branching pattern of our arteries. And we also note that resting state functional connectivity may express a certain amount of vascular connectivity in addition to functional connectivity that has been well studied. Thank you for your time, and thank you to the SNEL Lab for all their help.